So this is page 209, question 28, and in this question we're trying to compare the difference in between if the genes are linked and then if the genes are not linked. So it's a very, very, very important question. Um, and if you can kind of get your head around this, you'll be well on the way when it comes to a higher level exam. You more than likely will see some type of a cross, whether it be a sex linked or a dihybrid cross on a higher level, more likely to be a monohybrid cross dealing with just one single trait on an ordinary level paper. So in the question here, question 28, we have an organism of genotype capital S, capital T, small t was crossed with one of genotype, all lowercase letters SSTT. Show the progeny genotypes that could be produced if the genes were linked and if the genes were not linked, and then in each case indicate the expected ratio of each genotype. So we're just working out a cross for each. It tells us the parents, so we'll actually just put in the genotype of the parents first, and then we have that written out there. We can compare back to it. So genotype of parents, we have capital S, lowercase s, capital T, and then lowercase t, and that's being crossed here with all lowercase letters S, S, T, T. Okay, so that's what we're looking at. Just really important that we know whether something is linked or else not linked. So part A, we're looking at if it's linked. Now, when they're linked, just to really quickly to sketch out a little chromosome diagram here for people. So it's important that we have our head around this. We only ever draw the chromosomes that are relevant to us in these chromosome diagrams. So this signifies one pair of chromosome diagrams. Along these chromosomes, like you have your pairs of alleles for different traits. We're looking at S's and T's this time. So the way it works out is your capital S is on one, the lowercase is on the other. Then we have our capital T on one and then lowercase t on the other. Now the relevancy of this is when our gametes form, one of these chromosomes goes into one allele, the other one, one of these chromosomes goes into one gamete, the other one of these chromosome pair goes into the other gamete. So it's very important that we actually understand that. We'll just sketch out very quickly our second chromosome pair here. So we have one obviously coming from the father's side, one from the mother's side. It's no harm just to do these little chromosome diagram sketches just to get your head around it. So we have small s, and then small t, small t. So we're back to the normal sequence then. So we have our genotype. The question doesn't actually tell us what the phenotypes are, so there's no need for us to draw them out. Capital S, capital T, small t. And then we're crossing that with small s, small t, small t. So remember, like we said before, the gametes which must have one S and one T in each, but in this case here they're linked, so we don't split them in the same way. So let's think about it. It's like we kind of split apart these chromosomes. One chromosome goes into one gamete, the other chromosome goes into the other gamete. So the only outcomes we have here, capital S, capital T, which is this side, and then small s, small t, which is this side here. So we only have two gametes being produced. We'll compare that in a second to what happens when they're not linked. On this side here, they're the same, so again we will get that split obviously, but there's only one type of gamete. It's going to be small s and then small t. Then we come together, we do our Punnett square. Let's try and keep it kind of tight here because it's saved on space. We have another question to fit in underneath. So capital S, capital T, and then small s, small t, small s, small t here. And we know ourselves at this stage, use a ruler, keep it neat and tidy as we can. So the outcomes then, combining our s's here, first of all, capital S, capital T, small t, and then we have small s, and then small t, small t. So we only have two possible outcomes here in terms of the progeny that are produced. It's a 50-50 ratio, so the ratio is 1%, like it's 50% capital S, capital T, small t, and then 50% small s, small t, small t. That's if they're linked. Now if we compare that here to if they're not linked. Now if they're linked, it simply means that they're on the same chromosome. So if they're not linked, then what we're looking at is that they're actually on different chromosomes. So not linked. It's the same parents here, by the way. We're not changing anything in that sense. So we'll do a little chromosome diagram again here. So sketch out our cell. So in this case, because they're not linked, it means they're on different pairs of chromosomes. So I'm going to draw in two pairs of chromosomes. The first parent that we had was capital S, capital T, small t. Again, we'll just draw in the genes. So they're not linked. That means they're on different pairs of chromosomes. So we put the S's on one, and then we put our T's on the other. When you're sketching these out, it doesn't really make any difference where you put them. Our second parent then, so just drawing out our cell, we have two pairs of chromosomes. So there's chromosome pair 1, and then we have chromosome pair 2 that we're looking at. So in this case we have small s, and then lowercase t. So we'll just put in our s's, and then we'll put in our t's here. Okay, so we're back to the normal system. So we have our genotypes. So our genotypes here, we have capital S, capital T, small t, and then we're crossing that with our lowercase letters, so small s, small t, small t. I'm just going to move the page up a little bit here just so we don't run out of room. Okay, so in terms of our gametes then, that's our next step. And the gametes that are produced, remember there's an S and a T in each, so when we look up here, this pair of gametes are going to split, and this pair here are going to split. You're going to get one pair from each gamete into each of the, one pair from each pair of chromosomes into each gamete. So there's a chance that you'll end up with a capital S and a capital T in one of the gametes. It's a capital S with a capital T. You could have that capital S pairing up with a small t, so capital S, small t. This small s can pair up with the capital T, or else you could have a small s with a small t. And then on the opposite side, then just looking at the other parent, this one's a lot more straightforward. So the only thing that can happen is these will split, as will these, but you must have an S and a T in each. So we have small s, small t. So same as always, just put a circle around these here, just to highlight them. We'll just draw in our little arrows just to show they're coming from there. From there, the next step in the process is our Punnett square. Punnett square, so I'm just going to draw these in first of all. So capital S, capital T, capital S, lowercase t, lowercase s, capital T, and then lowercase s, lowercase t. And just always take your time with these letters, because if you do get them wrong, it will mess up your entire question. On the other side then we have a lowercase s and a lowercase t. So let's get my ruler again here. We'll just fill in all our lines. Always make an effort just to keep these lines in the right place, just because as an examiner it just makes things so much easier. Just when you can see where the student was, shows your kind of diligence really. Combining these then, we have capital S, lowercase s, capital T, lowercase t, capital S, lowercase s, and then lowercase t, lowercase s, capital T, lowercase t, and then the last one then, lowercase s, lowercase t, lowercase t. Now, the thing to look here is, instead of, when we compare this to the actual genotypes produced up above here, in the ones up above we only have 2 outcomes. In the ones down below here we have 4. Now the ratio of these is still 1:1:1:1, so they're the same in terms of they all carry equal value, but it's 25%. So 25%, 25%, 25%, 25%. Compare that to our 50-50 that we have up above here. The thing to get from this is if they are linked, which they are in this question for part A and not for part B, there's less variation out there. And then if we look down here, obviously when there's 4 different outcomes, we have more variation. Now, let's think about this as if the S's and the T's, if they actually represented something. So let's say the T's represented tallness, and let's say the S's represent the color of the leaves, or say the color of the flowers, and makes things easier. So let's say the S represented a silver, and the dominant T represented a tall. So in this case here, all these are going to be tall and silver. These ones here, they're lowercase t, so they're going to be short. And then let's say that the lowercase s represented black. So these are all going to be short and black. So tall and silver, short and black. That's all you have. But down here you'd have tall and silver. You would have silver and short. Then you could have black and tall, and then here you could have black and short. So you actually get to mix up the different traits, as opposed to just having your silver and tall always linked, and then your short and black always linked. So it's just a case of being careful with them, understanding our chromosome diagrams and how they split, and then just filling in the correct gametes and just taking your time with your Punnett square and making sure you draw it out correctly.